Hello, everyone. Am I audible? Can you hear
Yeah,
me? Okay, thank you. So, good morning, good afternoon, good evening, everyone joining with us from different parts of the world. Myself, Ashmi Rahman, I'm a final year medical undergraduate and founder and lead of Quantum Medicine Nexus Scholar Forum. And we are truly excited to conduct this section. This is especially for the scholars who are interested in brain science, particularly computational neuroscience. And why this is exciting? Because we have scholars from different academic and professional background. MBBS students, students doing MSc neurotechnology, researchers working with BCI, and all the scholars who has a different project ideas, which is very interesting for us. And we also hope we can bridge diverse perspective altogether. So this community, good morning, Deepshika. So this community, actually, there is a potential to form this as a solid interdisciplinary research team doing brain science, right? So we are looking forward to it. So after this section, what is my personal suggestion is you need to translate whatever knowledge and wisdom you're going to get from this section into real world project ideas. So, our community, who stands for interdisciplinary learning and research, what we believe is in order to study something complex, first we need to I mean, the effective way to study something is by doing it. For example, we need to convert the knowledge into its practical form, right? So, our community will support you for doing this, and we love to support you. So, for example, if you get a clarity on what are the research problems where our scientists deliberately attempting to resolve, that where that in that wisdom itself, like you will get an idea where to focus on, like what papers to work on. So you will get an idea actually what are the skill set that you need to attain to work on that project. And if you can be more precise, that will also give you an insight like what extent you have to study a particular skill in order to work on that project. So if we can, that precision actually give us more efficient output. Okay, so in our community, what basically we are doing is we are helping each other and discuss what is the next step you can take if you find any difficulty in doing projects. And we are actually reaching out to us like mentors so we can contribute each other for the further endeavors. Okay, so for the feasibility of this section, we are organizing this into three parts. First section, we focus on medical students who are very new to neuroscience, computational neuroscience, from zero to beginner level. So here we will discuss what is exactly computational neuroscience is, why we need mathematics and computation to understand brain, and how it is significant, what it's different from the traditional neuroscience, and what is actually happening in this field, and what are the foundational skill set that we need to attain to work on a project, and also like how one can be like eligible to apply for neuroscience lab, and how to approach lab, and how to approach a mentor. So we are expecting such kind of questions from you. And second section is for the young researchers or the students who has a prior knowledge in mathematics and Python. So kindly ask questions, that specific questions that can lead you to your projects. Okay, so and the third section is for the brain enthusiast. Uh, the academic background doesn't matter here. So you can either unmute yourself to speak, or you can put it in the chat box, and we will read it for you. So by the end of the section, we hope this section will give you an insight like what is that one necessary step you have to take to clear your path. Okay, so before asking, just keep this in mind that you are taking your first step for your first project. Okay, so before wrapping it up, I want to inform you one exciting news. There is a 10 days summer camp is coming up, organized by the Neurogati team. And we are so fortunate that this program is led by our professor, so we can ask him more about it. Right. So the actually for us, the success of this section is not just your participation, but seeing you to taking your steps to move further forward. Okay. So from our side, we are providing a community where you can discuss your project ideas. Just Just work on, just have a project idea, just have a team, collaborate, and working on that project. Okay, and we will help you as per our capacity. So, with all the grace and happiness, I would like to welcome our honorable, dearest Professor Srinivasa Chakravarthy to share his insight with us. He's from IIT Madras, Department of Biotechnology, and he's also director of Neuroscience, Computational Neuroscience Lab. So welcome, sir. Welcome, sir. And thank you so much be here and spending time with us. Over to you, sir.
Thank you very much, Ashmi, and it's a kind introduction. And I really appreciate the initiative you're taking to spread awareness about this field among medical community, medical students, medical professionals. Because I think this field has a lot of relevance to medical practice. Because usually engineering and medicine, they're like parallel tracks, and there is not much interaction. Only of late that interaction is growing in the area of medical devices. But otherwise, what I feel is there's a lot of exchange of ideas and knowledge that can happen, even when it comes to fundamental principles of operation of physiological systems, not just when it comes to devices. And that's where there is a lot of scope for collaboration, interaction between the medical community and engineering community in the field of neuroscience. So that's why I've titled this talk as Computational Neuroscience and Why Doctors Should Care. And so basically why it is so relevant to the medical practitioners. So if you just take a quick look at how medicine is developing of late, particularly neurology or neuropsychiatry, right? Earlier, people used to, when a patient comes, the doctor would ask the patient to, I don't know, stick the tongue out or do a knee reflex and very simple neurological testing, right? But now everything has become very high tech. You are made to take some blood work at all sorts of molecular parameters or MRI, CT, and so on, and you take all this data. And with increasing influence of AI in medical practice, a doctor doesn't even have to see the patient, get all the data and feed it into some AI platform, and then it will give you the diagnosis and even prescribe treatment and medication. So that's how it is evolving these days. So all this, I'm not a big supporter of this kind of a very impersonal approach to medical practice, but anyway, that's what is happening. And it's all driven by computer science and mathematics and all that, because all these tools, the foundation of all these tools is mathematics and computer science. So there is an unfortunate split, right? Now this is not only in India but in everywhere in the world. Biology and medicine. After your high school, if you hate mathematics, you'll go towards biology and medicine, or if you hate biology because it's descriptive, memorize a lot of facts, and if you can't memorize that. And if you like math, you go towards physics and engineering. So it's a very unfortunate kind of a split happening very early on. But ideally, what should happen is the two trends should continue, because there's a lot of scope for mathematics in biology, and biology can also inspire a lot of engineering, because nature is an excellent engineer, right? The kind of engineering examples that you find in nature, people are trying to imitate them. Like, for example, the glue that you find under the pads of some of these lizards, right? The people are trying to crack that material. And even more simpler examples like, fish in inspiring submarine, and birds inspiring flight, and so on and so forth, right? So there's a lot of inspiration that engineers get from biological systems, which are instantiations of excellent engineering. So there's a lot of crosstalk that is potential and possible between these two fields, and this kind of a separation is not at all healthy. So where that you find a gap, right, in the way medicine approaches systems, physiological systems, especially brain, is when it comes to describing function. So let me give a small example. So let us say you ask a physicist or an engineer, how does a calling bell work? The traditional calling bell, right? So there is there's kind of a magnetic material and that U-shaped rod. Around that you wind a kind of metal coil. When you press the switch, current goes to the coil. The coil gets magnetized. So magnetic field flows through to the rod, and that attracts the whole rod to the metal bar. So it strikes it, produces a sound. When it moves forward to strike that metal rod, the circuit breaks. So the spring pulls back the coil, right? And therefore current flows again. So therefore the magnetic field appears again. So it moves towards a rod again, hits it again, makes a sound again. This happens repeatedly, and you produce a buzzing sound. This is how it works, right? Now, in the same manner, right? If you ask somebody, how does Alzheimer's work? Or forget about disease, how does hippocampus work? You just say hippocampus stores memory, right? That's all. But exactly how does it store memory? Because if somebody asks a computer engineer, how does a hard disk store memories? They'll give you a very detailed explanation of what is special about a hard disk, what is a magnetic material, and how does it store information? Information in the form of binary information, zeros and ones. What are zeros and ones in a magnetic material? Then they tell you about the magnetic spins and how they can be oriented one way or the other. One way is zero, other way is opposite way is one. So you will find these stable clusters of magnetic particles, right, which are oriented one way or other direction. So each cluster represents one binary bit, zero or one. And if you want to write information, a magnetic head goes over the magnetic disk and writes this by manipulating the spin of these magnetic particles. So that a very detailed explanation can be given. The more detail you want, I mean, as much detail you want, an engineer will be able to give all the necessary detail. Right. Whereas when it comes to neuroscience, somebody says, asks, how does hippocampus work? We don't know. It just stores memory. That's all, like magic. Right. Whereas that should not be the case. We should be able to go deep and explain the operation just like you explain operation of a machine in engineering. Whereas exactly how do we do this in neurology today? We just also, if you're dealing with a disease, you describe symptoms, you localize lesions. Sometimes lesions are like, gross areas in the brain, like, like Broca's aphasia, there's a lesion in the Broca's area, etc. Or if you are more sophisticated, you talk in molecular terms, you can talk about alpha-synuclein aggregation or, tau beta tangles or, whatever. Right? So, and then we classify disorders. Okay, fine. But after the treatment, so treatment will be typically in some cases either you have the three broad categories, right? Medication, surgery, and physiotherapy, or some cognitive therapy. We'll talk about that later. But where is the mechanism? You make these big jumps, right? It's highly correlational. This symptom means this disease, and you'll also find this lesion means this disease, or this molecular development means this disease, right? And therefore, this is the treatment. And then you'll have some kind of a flow chart, the decision tree. Right? But in all this, there is hardly any consideration of a mechanism. You don't even ask for a mechanism. People don't even look at you as if you're an idiot if you ask, what is a mechanism? How does it work in the brain? For example, let's take Parkinson's disease. So we all know that Parkinson's occurs because of deficiency of dopamine. There is loss of cells in substantia nigra which release dopamine. So there are dopaminergic cells. So when these cells die, this chemical is going down in the brain, and therefore you see all these symptoms. But exactly how? Why does loss of dopamine cells, which causes dopamine deficiency, obviously, result in creation of tremor and rigidity, bradykinesia, speech problems, even autonomic problems, cognition, working memory, decision making. Why do all these things occur? Just because a very tiny part of the brain, the substantia nigra, is losing its cells. Okay, can you explain mechanistically? Usually, I mean, the textbook doesn't even talk about it. At the best, if you open the textbook, it will show you the direct pathway, indirect pathway, and this, dopamine is going down. Therefore, this pathway has become more overactive. So, under this in such vague terms, that's all. You really don't. In fact, if you ask some pointed question, take it seriously. It's really not clear how that kind of a very sketchy, vague picture can account for all this rich variation of, rich diversity of symptoms that you see. In fact, I had a very interesting experience some years ago. This was long ago when my lab was just beginning to just do research on basal ganglia. So I went for one of these DBT or DST kind of project presentations. Next to me, there was a neurosurgeon. So I asked him what he does, and he explained what he was doing, what he was proposing in that project. Then he asked me what I am doing. I told him I'm doing research on basal ganglia. We are trying to model basal ganglia. He said, "What is it to do research on basal ganglia?" I said, "What do you mean?" He said, "Isn't it all known?" What is? I said, "What is known?" Then he told me about this picture that is in standard textbooks, right? Direct pathway, indirect pathway, etc. I said, "How do you even know that it is correct? Have you ever verified it yourself?" Right. Then he looked at me like with a blank look. Yeah, I mean, the question didn't even arise to him. He thought whatever textbook is saying is correct, and then that's all there is to it. But is in science, you don't accept anything as a given thing unless you verify it yourself, right? So now even a very basic question we can ask is, how can you even verify whatever the textbook is saying about basal ganglia function? It's so sketchy, so high level and vague. It's a big research question in itself about, right, as to how to verify that description of basal ganglia function. So you see, there's a two contrasting approaches to the field, right? One is medical, one is engineering. In engineering, you talk about models and mechanisms, and then you talk about control. In medicine, you just look at, make observations, look at identify symptoms, then put the symptoms into clusters and you classify them and associate them with some kind of make a diagnosis and link it to some disease. And you localize this part of the brain is the cause of the problem. It's not that simple, right? This localization type of description of brain function is actually very misleading. People have written extensively about it. That is a very bad way of talking about brain function and brain pathology, but it's still very common. All the literature is saying that's not very good. Okay, so if you look at the main problem that I have been seeing for a very long time, but so I didn't have really, in-depth discussions with medical, experts, but whoever I know work closely, they understand this very well. So let us look at all that we do in terms of characterizing uh, brain or disease in medicine, right, into these compartments. So I'll say this microscopic level, macroscopic level. Because when you're studying brain, you can study it at multiple levels. You can study at molecular level in terms of genetics and RNA and mRNA, tRNA, proteins, and that's a very low level. Then you have cell level, right? And then you have small circuits of cells, what are called local circuits in neuroscience. And then you have a whole region. You can study amygdala, you can study putamen, right? You can study, I don't know, zona incerta, a very, very specific small part of the brain. Or you can study a whole subsystem. You can study visual system, you can study limbic system, or the somatosensory system, and so on. That's right. So you have these multiple levels. So let us broadly divide this into microscopic and macroscopic. At microscopic, you have all this blood work, genetic testing, PCR, histopathology, immunopathology. This is the kind of thing which is very familiar. In first year itself, we have so much of biochemistry and pharma, and all. You are very familiar with this. At macroscopic level, you have two things: structure and function. How do you study structure using MRI, CT, angiography, anatomical studies, biopsy, etc., etc. And when it comes to therapy, you do surgery, take out tissue. When it comes to function, what do you do? EEG, TMS, FNETs, MRI. These are all tools to probe, tools to measure, tools to manipulate, to have effect on brain. But when it comes to explaining brain function, that's where there is a huge gap. Medical textbooks hardly even talk about it. Right? How does thalamus work? Thalamus is described as a relay unit. Relay, what does a relay mean? Signal goes from the peripheries to thalamus, and then it picks up the signal and amplifies it again, sends it outwards further on to the cortex. That's what is called a relay. That's a gross simplification of thalamic function, right? So like that, there's just some random hand-wavy explanations because I think function is very deeply kind of ignored, and that's where I think there's a huge gap. And we need to understand function properly. And I think that language is language of mathematics and computational neuroscience. This is a big kind of a weak spot, I think, in medicine. If this can be strengthened with all these developments in computational neuroscience, then you will be able to interpret even these tools and their outcomes in a much better way. And that should also positively influence your therapies. So now, one more thing that happens in medical diagnosis is traditionally disease is, the understanding of disease is dependent on this idea of germ theory of disease, right? And behind all disease, traditionally we would imagine that there is some kind of a germ, a pathogen. And so that pathogen affects some tissue, some body parts, it spreads, and that produces some symptoms. And these symptoms are characteristic to that germ. So your job is to identify the germ by looking at the symptoms. And then your therapy or intervention is in terms of a drug, a medication, which will kill that germ. And then you'll be whole again. So this is a kind of a very simplistic approach to a germ theory of disease, right? How it, in medicine. But thing is, when it comes to brain disorders, a lot of major brain disorders, there is no obvious germ that you can pinpoint. Because let's say in infectious disease, encephalitis, there's a germ, so you have to kill it. But otherwise, things like schizophrenia, Parkinson's, Alzheimer's, depression, bipolar, epilepsy, there is no obvious germ, single germ or molecule that you can hit and somehow achieve cure. Right? There is a general breakdown of this very complex network, just like a car breaks down. There's no germ for a car. So there's just like a car or a vehicle or a complex machine breaks down. And there is typically certain modes of breakdown, the patterns of breaking down. I have to identify these modes of breaking down of the brain and then figure out how to intervene in this. So one thing, it looks very staggering and mind-boggling if you look at brain as a complex network and how do you fix it. But nice thing about brain is it will train itself, it will reorganize by itself. The only thing is how do you enable it to reorganize so that it will become whole or it will try to overcome the defects caused by the disorder. That itself is a big problem. It's not, although brain can in principle reorganize itself, it's not easy how to achieve that reorganization, retraining in a given subject, in a given patient. That's a huge challenge. Now, every mature science, especially the way we look at it from physics and engineering, right? Every science, take any science, like in rocket science, people say rocket science is a difficult science, right? But we are able to send probes to moon and Mars and all that. It's very with textbook precision. How are we able to do it? Okay, that's because there is a body of mathematics which is linked to physics that governs that entire field, and all the technology rides on top of that mathematics. It's not done by hit and trial. In the early days of a field, you do hit and trial. Like take any field, you start with observations. Let's say astronomy, or you start with observation. You look at stars, their positions, the planets, their positions, how they move through the year, right? And then make charts, and that's how you begin. The field begins. But after some time, you identify patterns in the data. That's the next stage. After some time, you fit equations and discover deep laws behind those data patterns, right? And that's what. So if you look at astronomy, Kepler has found these deep patterns in planetary motion, the Kepler's laws. And Newton came along and discovered, oh, there is a law of gravitation, right, operating in all the planets, and that's why they are moving like this. That's why you have all these three laws of planetary motion, Kepler's laws. Then once Newton discovered the laws of gravity, it became a universal law. It will work in any solar system. Whereas previous data and all that is confined to our solar system because that's what we are able to observe. But once you have a deep light, gets generalized immensely far beyond the instantiation from which those equations were derived originally. Okay, so that's how everything works in engineering, right? You write in engineering, there's circuit theory laws, civil engineering, structural engineering laws, and so on and so forth, right? But whereas in biology, we tend to constantly observe and collect observations, create mountains of observations. And with the current era of AI and big data, what is happening is because you have a lot of technology, you have funding available. You can keep on making observations, keep collecting data, keep collecting a lot of fMRI data, a lot of MRI data, mountains of this data. And then hopefully we'll figure out. But point is, the principles and the underlying laws are different from this data. We're supposed to extract these laws from the principles, but not just stay at the level of the data itself. I mean, so uh, the other thing is even in medicine, like in other organs are better, we are better off when it comes to the way we deal with other organs, like because they are easy to understand. Heart is a pump, right? And lung is a gas exchange system. You take in oxygen and send out carbon dioxide. Kidney is a filtration device. I mean, it's a very simple metaphor, engineering metaphor that you can apply this to that organ and at least you get some hang of it. Right? Muscles are physical actuators, etc., etc. So, but whereas when it comes to brain, it's a huge complex network and all these simple metaphors break down. You need to have a more subtle, more sophisticated language, which is the language of mathematics. So because of all these difficulties, what I just want to give a kind of a more dramatic, metaphorical illustration. Right. I don't know, for me, brain looks like a big cave, like a scary cave. Black box. People talk about black box. It's like a big black cave. And you stand at the mouth of the cave and peer into it. Oh, what is there inside? Oh man, there is something there. Keep guessing, right? And then you shout at it and you get echoes. Okay, those echoes are your EEG and symptoms and MRI scans, and, right? And but it's all from outside. Whereas, can we venture to go deep into the cave, walk around, look at the entire cave corridor system? How deep is it? Right? Does it branch out? Right? Can we, which corridor takes where? And so, like the way we would explore valiantly a cave system, can we explore the brain with all these tools, but not just stop short at collecting data, but try to get insight into what are the underlying laws behind all these mountains of data. So if we do that, I think we might find that brain is actually quite simple. Data is very complicated, is no doubt. But behind all that complex data, you might find that brain works on a handful of principles, because that's what you see again and again in engineering. Even the most complex system, take a Boeing 787 or a skyscraper with hundreds floors, right? The operating principle, the fundamental laws will be only economical in a few handful of laws. That's what you will read in your textbooks of civil engineering or I don't know, like electrical engineering or whatever. There will be only a handful of laws, but when you apply them in detail to a real world system, that looks complicated. But laws themselves are very simple, and everywhere you find the same laws in operation. That's what science has repeatedly confirmed uh, in its dealing with physical systems. They might look very complicated, but you actually understand it, and at the end of that whole exercise, you will find that it's very simple. There are only 3 laws in operation, 5 laws in operation. Everything is coming from those 5 laws. So I strongly believe that even brain can be reduced to those handful of laws, but requires some research and requires a little shift in orientation from this completely data obsessed approach to like in science, like in physics and engineering, more a principle centric approach. Right? And people often compare brain and a computer, because you always, when you study a system, you always try to connect it with something that you already know. Because brain is something like an unknown thing, very mysterious, unknown, mystical kind of a object. So we'll try to compare it with something that we know, and this is like this is like this. And try to work out associations. So people compare brain with the computer, but actually it's quite a misleading metaphor. Because, for example, in a computer you have a centralized clock, right, going at some gigahertz or something. Whereas in brain, there's no centralized clock. And the computer is digital, brain is not digital. It has works through these spikes. So anyway, so it's a superficial analogy, but if you take it seriously, it doesn't hold any water. So now let us talk about what is computational neuroscience. Like any computational science, computational neuroscience is a branch of science which describes brain in mathematical and computational terms. That is, you write down the mathematical equations to various aspects of brain function. And because very often these equations are so complicated, so earlier in your high school mathematics, if you have an equation, you can solve it and say x is equal to some a plus b or so whatever. But the equations that you use in neuroscience are so complicated, you may not be able to solve the equations just mathematically. So you simulate those equations, you write programs, and simulate it on a computer, and then see what will happen. For example, so field experiencing explosive growth. You can say in a way the field has begun in the 40s, right, or it has really taken off in the 50s and 60s. In the 80s, there was a huge kind of revolution that has taken place called the so-called connectionist revolution. And in the 2000s, the first decade of the millennium, right, the AI has really taken off. So as around same time, there's a lot of funding that went into neuroscience and computational neuroscience. For example, in the last decade alone, there was a huge project in Europe called the Human Brain Project with a budget of 1.2 billion euros, and about 100 or 200 million euros of that project went into building this very complicated whole brain computation model. They were able to, I think, build like whole brain of a rat or a mouse. Right, so that's the kind of funding that went into so it's the field has been experiencing huge growth. And there are a lot of conferences. I'm just showing you like sample 2, 3 conferences, but I mean, dozens of conferences all over the world. And we ourselves are organizing one conference, this summer. And lots of text literature is just exploding. I mean, I'm just showing you 2 pictures just for sake of concreteness. And journals, again, a lot of journals. Neuroimage for neuroimaging, cognitive neurodynamics. very old journals like Biological Cybernetics. Yeah, so it's a huge set of journals. Now, the goal of the whole field is to understand how brain works from many points of view. So because brain has many facets, you want to understand the electrical activity in this network, huge network. You can think of it as a static network, just a simplification, because if you want to describe brain, its entirety, all its aspects, it's too complicated. So you make assumptions. You look at it as simply a static network, and then you do a lot of stuff with it. Or you can think of it as a dynamical network, things changing through time, and then you can do a lot of things with it. Then math becomes very complicated if you imagine it as a dynamical network. You can operate at gross scale in terms of just regions in the brain. You can operate at microscopic scale in terms of single neurons, or what happens inside a synapse between two connections. People even study at such a low level that so there is something called a dendritic spine. Dendrites have little blips called dendritic spines, and that's where the synapse comes and makes a contact. And people study about the molecular flows of molecules within the little dendritic spine. Okay, so you can go to very high level to very low level. That's a huge field. A lot of work is going on. Now I'll give a quick overview of some of the things that you can do because field is so big, the many methods. I'll just give you so that you can grasp something, take something home at the end of this talk. I'll just give you a couple of examples. So think of brain as a network of neurons, and each neuron is an electrical circuit. It's an electrically active entity, right? Any cell in the body will have a voltage difference between inside and outside. But what is special about neuron is this voltage that is there inside is not a constant value. It keeps fluctuating as if there is it's conveying some signal. It's trying to say something. So a neuron can be in two states. In resting state, its voltage is more or less constant. It's about let's say minus 70, minus 60, minus 80 millivolts. And when the neuron gets into an excited state under certain conditions, this voltage shows rapid fluctuation, as you can see on the right side of the slide. Those rapid fluctuations, the sharp spikes, they are called action potentials. So this is the language by which one neuron talks to another neuron. And these signals are communicated to other neurons and they pick it up and they respond to it. And they can also get into excited state because of this. And this excitation sort of, the excitement, spreads over the brain. So if you want to think of the simplest mathematical model of a neuron, right, how can you do it? It should simply have at least, right, it should be able to capture a resting state and an excited state. So when neuron is in resting state, how will it go to the excited state? By receiving inputs from other people. Suppose, to take a human example, suppose somebody is not very excited about computational neuroscience, right? They're in a resting state as far as computational neuroscience is concerned. Then somebody like Ashmi will go and poke that person. Look, there is a field like this, and you should go learn more about it. There are workshops, learn more about it. Then they get into excited state. So what Ashmi does to other people is like input current, right? And then that person goes from resting state, excited state, as far as this field is concerned, this topic is concerned. So that's what happens in the brain. So now, so this excitation spreads from neuron to neuron via the connections or the synapses. And synapses, look at the picture of that, a graphical reconstruction in the slide. There are two sides to synapse. So one end is what is called a presynaptic terminal, that is before the synapse. Other end is called the postsynaptic terminal, that is after the synapse. And the presynaptic terminal, when the terminal receives a signal from neuron A, right, that causes release of a chemical called neurotransmitter. And this little chemical, a bunch of a few thousand molecules of that chemical get released, and then these molecules go to the other side. They land on the postsynaptic terminal, act on certain receptors, and produce an electrical change on the postsynaptic side. So basically what the synapse does is it takes the electrical signal coming from the presynaptic side and does some chemistry with it. And because of this, it gets converted into an electrical signal on the postsynaptic side. On the presynaptic side, the electrical signals are in the form of these action potentials, these sharp spikes. On the postsynaptic side, the electrical signals are not like action potentials. They look smoother, right, like what I'm showing here. And these are called postsynaptic potentials. So the meaning is quite obvious. The only thing is these postsynaptic potentials can be a positive wave or a negative wave, right? So that means the voltage can go up above the resting potential of minus 70, or it can go down. So depending upon whether it goes up or goes down, you think of synapses as having two categories, right? So there is a excitatory synapse where the postsynaptic potential goes above the baseline. Therefore, it is called the excitatory postsynaptic potential. Then there are inhibitory synapses in which the postsynaptic potential becomes negative, less than its normal value. And that it is called the inhibitory postsynaptic potential. And whether a synapse is negative or positive, inhibitory or excitatory, depends on a lot of the biology that happens inside a synapse. And this is not the context for it. I'll maybe describe it in detail in some other talk. So what a neuron is doing is take all these signals, the positive signals coming from other neurons via excitatory synapses, the negative signals coming from other neurons via inhibitory synapses, it combines them all and it makes a decision. Okay, I'm getting a lot of positives and only a few negatives, so maybe I should also say positive, and then it goes up, it gets excited. Or another neuron gets a lot of negative signals, only a few positive signals, it adds up all of them. It says, no, 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 it's not looking very good. I'm getting a lot of negative signals. Maybe I'll stay negative. That is, stay in the resting state. So like this, think of neurons as making decisions. I mean, we do this kind of thing also, like, suppose you want to decide, right, whether I should go abroad or not. I talk to 10 people. Out of 10 people, 8 people say, yeah, it's a very good thing, go abroad for higher studies. Then your decision about that goes from 0 to 1. Or you talk to 10 people, 8 of them say, no, 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 don't go, it's very bad right now, wars are going on. Then you're initially ambivalent, but after that you'll stay in negative. You don't. You take a no decision. So neurons are also doing something similar, but at the level of single cells. Now you can connect these neurons into networks. This is all done mathematically, and then you get a neural network, right? And this is what these are the kinds of neural networks you use these days in AI. These are called, for example, deep neural networks. When the neural network has lots of layers and has a large complex neural network, you call it a deep neural network. They take some input and produce some output. And you see that the neurons are connected by connections. And these are similar to synapses in the brain. The connection in the neural network and the synapse in the brain, they're similar. They're one represents the other. Okay, now these networks can be trained to do something, right? How is it trained? Let's say you want to make the network recognize faces. So you have 10 different kinds of faces it has to recognize, who is who. So give the picture of the face and then it goes to the network and then produces some output. Initially, when the network is initially it's very naive, it doesn't know anything. So all the connections are all random, right? So then it gives some wrong answer. Then you tell it, no, 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 this answer is wrong. This is the correct answer, and give the feedback. And then it takes that error in the feedback and then feeds the error back into the neural network and adjusts all the connections a little bit so that next time around when you give it the same picture, it will recognize that person in the picture with slightly higher accuracy. And this goes on and on. You do this over and over many times, hundreds of times, thousands of times, over all the pictures. And the network learns to recognize all the faces correctly. And in fact, this is how we also learn things in the world, right? I mean, look at this child trying to understand something about that pump by error feedback. So that's how you can train a neural network. This is a simple illustration of how a neural network is learning to recognize handwritten numbers. So with that, now let me apply this to a concrete system in the brain, the visual system, because what we are looking at is a visual function, right? How to recognize pictures. So you all know from your, I think, first year anatomy, right? How does the visual system, what is the organization of the visual system? So eye captures the image, right? Because of the optics, it will get inverted in the lens. And that goes by optic nerve to the first stopover, which is the thalamus. Within thalamus, there is a place called lateral geniculate nucleus where the signal goes. And within lateral geniculate nucleus, there are a whole bunch of layers of neurons. And then the signal goes through all those layers and goes out of thalamus and goes to the first cortical stopover, which is the primary visual cortex or B1. Okay, so there the image is seriously distorted, but fine, we're able to somehow magically see the world pretty reliably. And after that, after primary visual cortex or B1, the information goes further along two pathways. I mean, two pathways is one story. People say that also there's a third pathway. It's again like most things in neuroscience, there's a lot of controversy and difference in opinion about most systems. That's why I'm saying there's a lot of research that still needs to be done. So there are two pathways, the dorsal pathway, which goes upwards, into posterior parietal and towards somatosensory cortices and all that. Then the ventral pathway which goes downwards towards the inferior temporal and ends in IT, inferior temporal area. So that is shown in the picture below, right? And all along these two pathways, there are intermediate regions which do different things. There is V5, for example, which processes motion, moving information in the image. V4, which processes color. And some of these areas don't even have simple definition, but there are a lot of intermediate areas, right? Now you want to find out, you want to understand how does the system work when I give an image, a picture of a face or something. How am I recognizing that? Right, this is a picture of I don't know some film star or something like that. Right. So for that, let us compare it with a neural network because neural network is a computational model of something that's happening in the brain. So I will say that this is a model called the convolutional neural network, which is very good at recognizing pictures like the visual system. And within this, there are lots of layers, and each layer has two-dimensional arrays of neurons, and they're connected. These networks can get very big. In fact, anybody who has had some training in AI, one of the first things that they will learn in the first course on AI is a convolutional neural network. Okay, it's very good, very powerful model. Okay, and so these things have been trained like very large networks. Sure, for example, NVIDIA company has trained a very large network with 150 hidden layers and like billions and billions of parameters and all that. This is a network which was able to drive a driverless car, right? Automatically looks at video coming from the camera feed of the car. It can drive and control steering angle. Okay, so these networks are so good that they're even able to beat humans at some of these tasks. For example, there's one famous data set called German traffic sign data set. These are photographs of traffic signs because if you have a driverless car, you should be able to follow traffic signs, right? Road signs, speed limit signs, those kinds of things. So some of these traffic signs are taken under such poor lighting, at let's say evening time or the atmosphere, the clouds. There's a lot of clouds and the sky is downcast and all that. So how do you recognize in such poor lighting conditions? So they compared humans with the performance of the network, and sometimes the network beat humans. So that is called superhuman performance. So at the level of behavior, that's input-output behavior, networks are sometimes able to beat humans. I mean, we know that even our computer, that a traditional computer, can beat humans on certain tasks. So if I give you like multiply two 10-digit numbers, I will take forever to do that multiplication. A computer will do it in a second. So that is fine. But what we always thought is there are certain tasks where humans are very good at and computers are not that good at. Like for example, recognizing pictures, when there's a lot of clutter and noise and all that. But even in such areas, now the AI systems are beating computers. In fact, AI systems are gone much further than what I'm talking about here. But anyways, this is a trend now. So now if you train a convolutional network on some image recognition task, and then if you go into the network and see what the neurons are responding to. So what you do is, so you present a picture and then look at the neuron and see when all this neuron is going high. What kind of an inputs does excite this neuron? This kind of experiments people do on real animals. So for example, the famous Nobel-winning work of Hubel and Wiesel at Harvard University in the 60s, right? What they have done is they present some simple patterns like a bar, like a rod or something like that, some angle. Okay, and then record from neurons in a monkey and a primate. And then see, they found that if only a rod of a specific angle, orientation, will excite that neuron. Right, so neurons in V1 have something called orientation sensitivity. They respond well to a certain optimal orientation of linear features, line-like features. And that's what is shown in this part of the figure. So V1 neurons respond to lines and of given orientation. Then what about the next layer in visual system, V2? So people have found that these neurons respond not to lines, but combinations of lines. It can be a combination of two lines like an angle, or a combination of three lines like a Y, right, etc., etc. And so on. If you go higher and higher, go all the way to inferior temporal area, neurons respond to complex images, like especially images of faces. The faces can be human faces, animal faces, but somehow neurons in IT area like faces. Now, what they found is even in a convolutional neural network, which is trained on all these artificial images, the neurons seem to again show a similar pattern. Lower layers respond to simple line-like features, higher layers respond to more complex features. Okay, that's very interesting because this is a very general abstract network. I didn't try to copy the visual system, part by part in the CN in the convolutional network. It's a very general network. The only thing we have done is train it on some kind of a broad image data set. And neurons seem to behave just like how neurons behave in the real brain. So this is very interesting because what that means is this neural network model, which we thought is abstract, is actually capturing something that's happening in the real brain. Okay, this is a very simple example of what kind of insights a neural network can give about brain's function. Now, let me go further. You can get a lot more than that. So what people have done, this is a nice review by Kell and McDermott. So what they've done is they've trained a neural network to recognize some images, and the same images were given to real humans and asked them to recognize those patterns. And while they are doing that task, this recognition task, their brains were scanned in an fMRI device. fMRI device gives you a functional understanding of the brain. It tells you which parts of the brain are activated when you're doing some task. So then what they found, so they got the activity levels of V1, V2, MT, V4, and all that stuff while the person is doing this task. And then the neural network is also trained on the same task. And then you can find out, right, when I give some this image, which part of the hidden layer is activated and so on and so forth. And they tried to predict the activity of a given brain area using activity of a given hidden layer in the artificial neural network, the CNN. And then what they found is the lower layers in the CNN behave a lot like the lower layers in the visual system. And the higher layers in CNN behave a lot like higher layers in the visual system. Which means there's a correspondence between the CNN and the visual system. It's an approximate correspondence, right? So it's not just at the level of input and output, even at the level of what is happening inside the network, level of neurons, there's a lot of correspondence between this artificial system and the real brain. Now let us go deeper into this. So that's an example of how brain works under normal conditions. Now, what about diseases? Like I said, disease in the brain is a case of network breakdown. There's a failure of the network. And there are models. So one thing I must tell you, computational neuroscience is still, there's a lot of research going on. We don't have like a final word on any of these disorders. You'll find immense amount of research material, research literature. But if you say, tell me finally, okay, well, how does Alzheimer's work and can I cure it? Means you don't have a definitive answer. We'll have lots of theories, a lot of papers, and so on. So it's still there's research that has to be done. But there's a lot of material already out there. So there are models of, so we ourselves have done work on models of stroke. We have done a lot of work on Parkinson's, modeling of Parkinson's. Then there is schizophrenia. We have proposed one model for bipolar disorder about 6-7 years ago. Then there are models of epilepsy, Alzheimer's, and so on. So let me take example of Parkinson's. Again, the Parkinson's occurs because of loss of cells in this tiny part, the substantia nigra. And so the thing is, these cells die in almost pretty much everybody. So only when the cell loss causes a certain percentage, you start seeing symptoms dramatically. That's when you say a diagnosis. But the thing is, this cell cluster is so deep, and then these cells form part of what is called the diffuse projection systems. So these cells send their fibers, these wires, diffusely over large, targets, distributed set of targets in the brain, and they have to drive signals on top of all those, like very complex and long highways in the brain, and which is very energy intensive. So these neurons are under very high metabolic stress. They're very hardworking. They're overworked. So therefore, they are under metabolic stress and they're likely to die. They're vulnerable to cell death. Only thing is, it happens a little bit more in some than others, and that's the problem with the disease. So once the symptoms start appearing, the symptoms can fluctuate over months and years, and medication effects are also quite variable. And deep brain stimulation, traditionally people would do put electrodes in the brain, typically in subthalamic nucleus or in GPI parts of the basal ganglia and electrically stimulated. So when medication stops working, people go for DBS as a last ditch, like a last resort. But of late, a couple of years ago, I was at this international conference. I had to give a keynote lecture. The conference is full of clinicians. A lot of them are from UK. And you know the main conclusion in that conference, they were all saying, look, don't wait for late stage to put DBS electrodes. The moment you diagnose, put DBS electrodes. I don't know. I don't understand the clinical world that much, but I think should we really should we really go for such a seriously invasive method right from the beginning? Okay, so what the model can do, we have done a lot of modeling. What the model can do is give a lot of insight into how the loss of cells in this tiny part can cause all these symptoms. So if you take a quick review of Parkinson's, I said it's because of loss of cells in SNC. The four cardinal symptoms: tremor, rigidity, postural abnormalities, and bradykinesia. And so thing is, in Parkinson's, because of loss of these cells, pretty much everything that brain does can be affected. There are four major domains of brain function, right? Sensory motor function, cognitive function, affective or emotional function related to limbic system, and finally autonomic function. In PD, all these four can be affected. In a given patient, some may be affected, some may be not. There's a lot of variability from patient to patient. But in this principle, all this can be affected because these cells are at such a high level in the hierarchy, right? That they influence all these different domains of brain function. In motor function alone, you can have problems in hand movements, reaching, handwriting, eye movements, gait, speech, posture. I mean, that's pretty much everything, all motor function. There's nothing left. Okay, so why does it happen? To understand that, one key thing we need to understand is the nature of dopamine. So dopamine is called the pleasure chemical, right, of the brain. It is released whenever you are happy. Like, if you take stimulants and opiates and drugs, even food is, we love to eat because food is enjoyable and it causes release of dopamine. And dopamine antagonists, that is, these are chemicals which attenuate the action of dopamine, we block the action of dopamine. It will also, in psychological terms, it will also suppress the pleasurable experience caused by whatever, all these things, right? In addition to just chemicals and drugs and food and all that, even presentation of beautiful faces and images of lovers. And lovers generally think that, the boy thinks that girl is the most beautiful girl in the world, and the girl thinks that boy is the most handsome boy in the world. I mean, that's why they're called lovers, right? So people have put lovers in scanners, fMRI scanners, and notice that the reward system, that is the part of the brain linked to dopamine function, it lights up. That's why, that indicates that they are in love. So similarly, monetary reward. So somebody gives you a huge cash, wad of cash, and you'll be very happy with it. So that's when you see the dopamine release occurs when that happens. So basically, what happens here is, what does it mean to get pleasure or pain, right? So in more formal psychological terms, you can talk about reward and punishment. So let us say you have, there's an agent or an animal, there's a stimulus and produce a response. And the response leads to reward. Next time it will continue to do the same thing under those conditions. The response leads to punishment. Next time it will avoid doing that, right, under the same conditions. So you see that this is called reinforcement learning. I'm putting it very simply, but there's a huge mathematical body of knowledge, which is a part of AI, modern AI, called reinforcement learning. And you see, for an agent to perform reinforcement learning, it needs to combine three things: stimulus information, response information, reward information. Now look at how basal ganglia is located inside the brain, right? It gets inputs from the cortex, from sensory cortex and motor cortex. Sensory cortex tells it about stimulus information. Motor cortex tells it about motor action, response information. And SNC, which projects to basal ganglia, tells it about reward information because it releases dopamine to basal ganglia. So this block, which is basal ganglia, is just like this block. All three things come together, and no wonder it's able to combine all this and now instruct the cortex, the motor and the planning areas of prefrontal. Okay, whatever you have done just now, it's great. Next time, go ahead and do the same thing when you get the stimulus. Or whatever you have done just now is not very good. Don't do it next time when you get this stimulus. So that is reinforcement learning. And you see how basal ganglia is well positioned in brain's anatomy to perform that kind of a function. So we have taken this task about 2005, 2006 and published a lot of papers and also a book which summarizes all our work on basal ganglia. And so then after that we began working on other systems like hippocampus. We've done a lot of work on spatial navigation, some work on visual system, and so on and so forth. Our long-term goal is my lab is to model the whole brain, which we call the mesobrain. The mesobrain is actually a kind of a slightly simplified essential picture of the brain, a mammalian brain. So in this, you have the sensory inputs on the right side, touch, sound, vision, and so on. And here you have primary cortex, secondary cortex, and so on and so forth. On the other side, you have motor outputs coming down, the descending pathways, right, as a musculoskeletal and speech and oculomotor and all these things. Between the two, you have this orange block, which is the planning and decision-making regions of the prefrontal. You have your dorsolateral prefrontal, anterior cingulate, and orbitofrontal cortex, and all these different parts of the prefrontal cortex. And then you have three parallel loops: cerebellum, basal ganglia, and hippocampus. Okay, so my lab is divided into teams. Each team works on one focused area, and such. And I've also written a book called Demystifying the Brain. The purpose of this book is just what I'm doing right now. That is, how do you communicate all these mathematical theories of the brain to somebody who doesn't know much mathematics, right? To a doctor or a biologist, how do you communicate this? This book is pretty. So I have even a full semester course based on this book. This book covers everything from history of neuroscience to theories of consciousness at the other end. Okay, and now let me talk a little bit about comp neuro in medicine. Where is it used in the clinic? Like I said, a lot of it is still at research stage, but some are used in clinic, for example, diagnostics. So now I'm saying I'm using AI and comp neuro terms almost interchangeably because AI for diagnostics in neuroscience is used already extensively for EEG, MRI, fMRI, so on and so forth. Neuromodulation, that is, neuromodulation is about brain stimulation. And this can be done at multiple levels. Like you can have very invasive ones like deep brain stimulation, or you can have more superficial stimulation, like you put electrodes on the scalp and stimulate. You can pass DC currents and AC currents and so on and so forth, or you can pass magnetic fields, magnetic pulses using a TMS. There's a lot of modeling done in this space also. Then rehab, to understand how we learn both cognitive function and cognitive rehab and motor rehab, a lot of models. And now one subcategory of rehab, which is becoming quite popular and where we are also quite active, is game-based rehab. That is, how do you design rehab based on gaming, right? Instead of just asking the patient to do repetitive exercise, how do you gamify it and design games so that it's naturally engaging to the patient? Because why is gaming important for rehab? Because gaming is enjoyable, which means it will activate the reward system. It will cause release of dopamine. And dopamine is a neuromodulator. It will induce neuroplasticity. That's why a lot of people say that gaming-based training is better because it will get you better plasticity. Then drug discovery, again, there's a lot of work. So traditionally, drug discovery would be simply, when people talk about modeling in drug discovery, people would take computational models to discover drug at molecular level. So once you have the hit on the drug target, right, people go and do molecular biology in the lab, in the wet lab. After that, you do animal studies and then human studies, human trials, and so on and so forth. But this route for drug development is very expensive. The latest figure somebody was telling me from a pharma company, it takes a couple of $1 billion. B for boy, right? Whereas the another school of thought is there's a whole field called computational neuropharmacology, where the approach is uh, do the entire thing in simulation first in a computer model. That is from molecular level to wet lab studies to animal trials to human, everything you do in a simulation first. And if you are convinced that that is working, only then you go for studies on the real wet lab and animals and all this. This will be a lot cheaper because the computer study can be done at the cost of a couple of $1 million or something, not 2 billion dollars. Okay, so therefore there's a lot of potential. Some applications are already being used. A lot of research is going on. But I feel if the future doctors join this journey at a very early stage, by the time they go out and begin to practice, right, they'll be at least familiar with these approaches. So that will help their own practice later. That's the whole idea. So a future doctor will deal with digital twins and predictive models and closed loop, neurofeedback, et cetera, et cetera. Neurofeedback for simply behavioral training, neurofeedback for deep brain stimulation. A lot of things are happening possible. So for this, medicos don't have to learn advanced mathematics. All that is done by engineers, and then they build these models. It's enough if the doctor understands what is this model essentially saying and how to use it. So you don't have to have in-depth understanding of the equations and all that in the model. Okay. So the take-home message is brain disorders are network failures. And verbal description not enough. Because even engineering, verbal description is not at all enough. After initial verbal description, you have to go to a mathematical description. That's why my students also, discuss something very quickly. I say, okay, fine. Now express whatever you're trying to say in mathematics in the language of mathematics. Only then we are able to understand what you're talking about. Otherwise, you're saying something, I'm saying something. It's chaos, right? So okay, and so building foundations, because Ashmi asked, like, how do you build foundations? If some of you would have gone through central syllabus, right, at +2 level, that's a great advantage because central syllabus has mathematics also. Otherwise, you would have left math in your 10th grade, must have been long ago. So some knowledge of mathematics, calculus, differential equations, and when it comes to physics, circuit theory would be useful. Electrochemistry, basic electrochemistry. Then slowly get into learn some AI tools because AI is a very nice side door entry into neuroscience. Because AI will have applications in other things, right? And it's very useful, so you can pick it up. And once you understand what is a network, how do you train it, and so on, those ideas can be quietly carried over into neuroscience to understand various neural phenomena, right? And then slowly you can pick up some basic ideas about what is a dynamical system, what is an attractor, what are oscillations, and right, what is synchronization. All these things you can pick up, as you go along. Because ideal thing would be to learn a little bit, then apply it and see that it's useful for your work. Then you get motivation to learn more. Otherwise, if you just learn for many years and then you find it's only a little bit of it useful, then it's not very beneficial. Then programming, yeah, basic Python, and after that now there are these AI tools which will let you even do the coding, right? But I think it's important to learn some principles of programming, some basic expertise in programming will be very useful. So then you can take, first short courses, like 3-day, 4-day courses, and longer term courses. In fact, I was talking to the HOD of neurology in Ramchandra, a good friend of mine. It would be nice to add courses like this in medical curriculum. But thing is, straight away, they may not be open to doing something like that. But what can be done is you can have courses outside the mainstream medical curriculum. And as more and more students take these kind of courses outside, then the medical administrators themselves might think that it's good to add them to the mainstream curriculum. That might as very likely to happen right over the next few years. So in my startup, we are having all these workshops. In the summer, we are having this huge workshop, a huge conference, international conference, and with about 30 plus speakers from all over the world, from US on one side, Australia on the other. Then we have abstract presentation. If anybody has any research even vaguely related to Comp Neuro, please feel free to submit abstracts. And we also have hands-on sessions where you can get in-depth training on one topic, right? About 6 such hands-on sessions. Then actually this last week we have been talking to some people from Bay Area, San Francisco area. There's some interest there, so we will be offering a 2-week program between June 1st to June 15th, mostly for high school kids in that area. But I mean, the classes will be offered in the evenings so that they can join, but anybody can join if the time is suitable for them. Right. So we want to offer a like a short workshop for medicos, like something like Comp Neuro for brain modeling for medicos or something like that. There's enough interest, and depending upon the convenience in terms of timing and schedule and all that, we can work out something, and we can plan to offer something like that. Okay, thank you. And now I'll take some questions.